So, in order for a lymphocyte to fully become activated, there are several signals that must be in place, chemical signals. This protects us from hyperactivation. And some of the things that can make this stimulation and activation occur, we call cytokines. Those are chemicals that are released by immune system cells. So if you take a look at this, it says here is a helper T cell. That's a type of T cell. And helper T cells have a T cell receptor that are going to be specific for an antigen. And here is a macrophage that has eaten a foreign invader, chopped it up and processed it, and put it on the surface via an MHC class II molecule. So a helper T cell must be presented with an antigen. It cannot bind free-floating antigens. And so the antigen-presenting cells are really important in this way, that they eat the foreign invaders and present the antigen to helper T cells. So the helper T cell binds, the macrophage has its displayed antigen, and that is considered the first signal. Once we get this binding between these two cells, okay, the macrophages will release cytokines that can bind to the helper T cell, and we call that co-stimulation. Co-stimulation ensures, kind of as a double check, like, yes, this is a foreign antigen, now you can become activated. There are other types of co-stimulatory molecules. If you take a look here, here's a helper T cell, here's a macrophage. Here are a couple of other surface molecules on the surface of helper T cells and macrophages. So if you look at this like reddish kind of thing, pink thing, this is a CD4 molecule on the surface of a helper T cell. Not only does the T cell receptor bind to the antigen, but the CD4 protein will actually also bind to the MHC class II molecule, checking to make sure that this is good, and it helps hold the cells together. Additionally, you'll see proteins can extend out of the macrophage and the helper T cell, this CD28. Protein can come out and bind to this B7, and that is further stimulation that yes, and checking that yes, this is a foreign antigen. So there's lots of signaling that occurs here. We get binding of the antigen, binding of the CD4 molecule to the macrophages, MHC class II molecule, and then binding of cell proteins. Not to mention, we had the cytokines being released from the macrophage and binding to the helper T cell. So lots of co-stimulation necessary. And again, this is to ensure that, yes, these cells are truly binding and recognizing foreign antigens. Okay. So like I said, in order for a helper T cell to become activated and then clone itself, it has to have an antigen presented to it. So let's take a look at this whole process, putting it together. Okay, here is a foreign antigen. Here's a macrophage that's going to eat that foreign antigen, chop it up, and put it on the surface of the cell via an MHC class II molecule. A helper T cell will come and bind to the processed antigen. The CD4 marker will come from the helper T cell. Again, this is a protein that sticks out of the surface of the helper T cell, bind to the MHC class II molecule. A CD28 is going to bind to the B7. This is called co-stimulation. Additionally, the macrophage will start to release cytokines. In this case, it's called interleukin 1. You don't have to memorize the name of interleukin 1. And you see that interleukin 1 will bind to receptors on the helper T cell. All of these things need to be in place for the helper T cell to truly become activated. Once the helper T cell becomes activated and is co-stimulated, then the helper T cell can start producing something called interleukin 2. Helper T cell releases interleukin 2, which will bind to a receptor on its own surface. Isn't that kind of funny? It kind of is a loop here. Interleukin 2 binds to its own receptor on the helper T cell surface, and now the helper T cell can clone itself. And so here we go. You see helper T cells can be stimulated to divide again. We can make more clones, or helper T cells can then help stimulate B cells or effector T cells. So the function of helper T cells, okay, if I was in class, I'd be writing stuff on this PowerPoint, right? Helper T cells that are activated help activate B cells and other T cells. So some people say that helper T cells are essentially like the sergeants or the generals of an attack, right? They're kind of activating everybody else and getting them ready for battle to fight. So that's what we're going to see in the next slide here. That in order for a B cell to fully become activated and then clone itself, it typically requires a helper T cell. So let's see what's going on here. It says, number one, before a B cell can be activated by a helper T cell, the B cell must endosynthesize phagocytosis and process the same antigen that activated the helper T cell. Okay, so remember B cells, guys, B cells also are antigen presenting cells. They're going to take a foreign entity, take it, bind to the B cell receptor, bring it in via phagocytosis, chop it up, and display it on the surface via class 2 molecule. The activated T cell that's already been activated by an antigen presenting cell comes and binds to that processed antigen, so they both have to be specific for this antigen. Okay, the helper T cell comes and binds to that antigen. We get CD4 binding. We get chemicals released from the helper T cell. And the helper T cell will stimulate the B cell to then divide by mitosis and cytokinesis and clone itself. Notice that some of the daughter cells will become what we say are effector cells. Okay, effector cells, I'm going to write this here so you can remember this. Okay, plasma cells are effector cells. Effector cells fight in the current infection. Some of the cells that are clones are going to become memory B cells. They will remember this antigen and can fight in future infections. Okay, plasma cells look different than these clones. They change quite a bit, and you'll see all this green stuff. They have a lot of rough ER in here, okay, because they produce antibodies like crazy, and that's what they start doing. They start secreting antibodies. In the next slide, we're going to discuss antibody-mediated immunity.